Hello, and welcome to today's webinar, Environmental Chemicals in Breast Developmental Timing. I'm Dr. Jill Oxley, and I'm a member of the board of Massachusetts Breast Cancer Coalition, and I'd like to thank you all for joining us today. I'm pleased to welcome today's speaker, Dr. Suzanne Fenton. Dr. Fenton is the group leader for reproductive endocrinology in the National Toxicology Program Laboratory. Her laboratory has published numerous manuscripts enhancing the methodology used in mammary gland assessment and determining early life chemical exposures that lead to persistent developmental changes in breast tissue, altered function, or disease susceptibility over the life course. She has received several awards from the National Institutes of Health and the EPA for her research on perfluorinated chemicals and endocrine disruptors. She is also a 2019 NIH Mentoring Award recipient. Dr. Fenton earned her MS and PhD from the University of Wisconsin-Madison in the Endocrinology and Reproductive Physiology program. Following her postdoctoral fellowship at the UNC Chapel Hill Lineberger Cancer Center, she led a research laboratory at the US EPA's Reproductive Toxicology Division for 11 years before she joined the National Institute of Environmental Health Sciences in October 2009. Dr. Fenton, welcome.
Thank you so much. And I just want to say happy anniversary to MBCC. 30 years is a long time. Hopefully you can keep it going. There's a lot of good work that has to be done. I want to talk today about some rejuvenated efforts in the field. And as you know, there's a new director at the National Cancer Institute. At the NIEHS, we have had numerous meetings with our grantees and with the NCI to sort of jumpstart some new ideas and new programs, some more thought process to how the environment is affecting breast cancer. And so I've been involved in those efforts. We should have a summary of our meetings that comes out soon. And when that happens, I can forward them to you, but they're not available for me to share right now. So one of the reasons that we have sort of a renewed effort and like meaningfulness right now is not only because there's a new director and a lot of things have changed in the administration and the feelings toward cancer research at a very high level, but also because breast cancer in young women does not seem to be slowing down. Breast cancer in young women is more prevalent than any kind of cancer in men. So as you can see here, these are very recent data showing that breast cancer is far and away more prevalent than any cancer in men. And we, funding-wise, that is not it's not equivalent. When we do studies on breast cancer and the environment, we use rodent models. And if you're not familiar with what that looks like or why we do it or any of that, I just wanted to quickly show you this picture that, in women, we have one set of breasts. In rodents, a rat has 6 sets of mammary glands and a mouse has 5 sets of mammary glands. So I can get quite a bit of tissue and we can expose them to the environmental contaminants of interest, and you can specifically then see the effects of those environmental exposures either on a slide that we have read by our pathologists or in mammary hole mounts, which you'll see quite a few pictures of in my talk because we find this to be a really useful way to evaluate lesions in the gland or early signs of disease. So mammary hole mount is really taking the entire gland and putting it on a slide, staining it, and then you can see it in 3 dimensions. So you can see all these branches down here. These are all the epithelial ducts of the gland. And we can tell when there's something wrong, like here at the bottom. So one of the important parts about this is that during development, there are a lot of similarities between rodents, rats and mice, and humans. So you can see that comparison shown here, where the rats and the mice are shown at the top two panels and the human is shown at the bottom. The ductal development that you see going into infancy and childhood looks slightly different, but is very similar regardless of which species you're looking at it in. In puberty, all three of those species develop what we call terminal end buds. Those are very sensitive to the effects of chemicals and especially to carcinogens. They're rapidly dividing cells, and you don't want terminal end buds to be exposed to a carcinogen because there is a link between carcinogen exposure, terminal end bud, numbers of terminal end buds in the gland. Another interesting component of doing this work in animals is that we can't tell the difference between female and male mammary gland development in the fetus. So here are shown the female and male mammary bud in a rat strain that we commonly use. And I can't tell the difference, so I doubt that you can. And it's interesting that there are very similar expression of receptors, very similar morphology in all ways at this stage. It's not until the male has a androgen surge during fetal development a little later than this that things start to change slightly. And really then at birth, if you look at the whole amounts of mammary glands, you can't really tell the difference. Oops, so sorry, I don't know how that happened. You really can't tell the difference between females and males here. And you can't tell the difference even when they're weaned, which is still before puberty. You can't tell the difference, but once puberty hits and the male's hormones go into high gear, then you can really tell the difference. And there's a stunting of mammary gland development in the male that never recovers. And then they are definitely different, and we can see the differences. One of the really nice uses of these whole mounts is to tell the difference in delayed versus accelerated development of the gland. So we always compare those to what the control looks like here in the middle. So this is what mammary gland development should look like in the animals under normal circumstances at the same age. If we see delayed development, like we have demonstrated for dioxin, PFOA, Genex, which I'm going to show you the data for in a minute, atrazine, which is a herbicide, and a mixture of flame retardants called PBDE. Those all cause either permanent or transient delays in development, whereas more estrogenic substances like the bisphenols, some of the nonylphenols, a lot of these things are found in plastics or coated products. They have more estrogen-like qualities to them, and they cause accelerated development. I'm going to show you an example of what that can lead to in a little bit. So this accelerated development and the delayed development can both have negative effects. In fact, EFSA, which is the European Food Safety Administration, has made a decision on these effects of BPA in the mammary gland, or these bisphenols in the mammary gland, that they consider these effects likely to cause effect and that the effects in experimental animals are relevant to what might happen in humans. So they've made the decision based on the science that the effects shown here could also happen in humans. One of the problems in the breast, and why we don't know more than we know already, is that there are a lot of endocrine disruptor targets in the breast and in the body, actually. So there are a lot of places that an endocrine disruptor could have an effect, even if it doesn't get to the breast, and to still affect the breast. So we know that all of these endocrine targets, the pituitary gland, thyroid, adrenal, liver, metabolism especially, ovary, placenta, pancreas, adipose tissue, salivary glands, all of these are targets for endocrine disruptors. Some of the things that might change and change how much is in the bloodstream and it gets to the breast are shown here. And so these can either have permanent developmental effects or maybe transient that can recover. Also, some EDCs may have potentially a carcinogenic effect, but most of them is more of a developmental change where you might have that it might change your susceptibility to cancer risk later in life if you were exposed to another hit or a carcinogen. Another reason for us not knowing more than we already know is that the breast is a really complicated tissue. So not only can it be affected by a lot of different endocrine sources, and in addition to the breast itself being affected in these 5 different cell types. So all 5 of these cell types shown here, the adipose tissue, the blood vessel type cells, stromal cells, inflammatory type cells, and the epithelium, which is the ductal and terminal end bud structures, they all can respond to endocrine disruptors. They respond in different ways, and it's really the way in which they respond and talk to the epithelium that matters. So we see changes not only in the epithelium, but in other cell types. And I'll show you some examples of that in a minute. But another problem that's happening in the breast, not only in young women, premenopausal women, potentially that develop breast cancer, but we're also seeing a shift in global breast developmental timing. So this is an example from a Scandinavian study showing on the top here a big gap between the early 1990 breast developmental timing. So this is the probability of having breast buds at a certain age, shown here on the bottom. So in the 90s, it was happening later. So say you want to say, what's the chance of 50% of the girls having breast buds? That would have been at about 11 years old in the 1990s. If you go to this blue line, which is the 2000s, just a decade later, you find that the answer is different. It's maybe 10 years old. So breast developmental timing is happening much earlier, but that doesn't go hand in hand with the timing of their first period or the probability of menarche at a certain age. That has not changed much, maybe a month or so. So there's something happening, potentially the environment, potentially increase in obesity status, which of course could also be affected by the environment, that's affecting the breast, specifically the breast, and not other targets within the body. So these windows of sensitivity, the gestational kind of breast bud period and the pubertal period when these terminal end buds are present, have been shown to be highly sensitive to chemical exposures. In fact, this is just a summary, but some of the many outcomes that have been reported due to chemical exposure in rodent studies and human studies, some of them, are either altered developmental programming of the gland, pubertal timing changes, inappropriate gender-specific characteristics, which has also happened with like gynecomastia in boys, can be altered by environmental exposures, especially some certain oils. We can also have extended presence of terminal end buds in the animal studies, which we know can lead to adverse outcomes later in life. And so some of this work has actually led to kind of pediatricians rewriting the book on precocious puberty, because precocious puberty isn't what it used to be. It's much earlier now, and it doesn't necessarily lead to pharmaceutical treatment as it used to. Sorry. So not only is there the similarity in development, but there's also a similarity in life stage susceptibility between rodents and humans. And that's kind of shown in like not only just the age or stage, but also what that might look like. So here, shown here. So when the mammary gland's exposed to environmental chemicals early in life, you may see that. And I'm so sorry about my dogs who think it's really funny that it's raining really hard outside right now. So the physiological outcomes can be seen in early development, in puberty, and these can manifest into lactational changes and to increases in cancer outcomes. And those have been reported for this large group of chemicals shown on the right. But if you think about how many chemicals are on the marketplace, this is a very small list. So I think it's a small list, even though it takes up the whole page. And most of these have not been assessed for their effects in women. We know a lot of these effects in animal models, but things like light at night and shift work has been recently reported in women. And from the work of Barbara Cohn and her colleagues, Mary Beth Terry, and several others, we know now that this may also be true in women, and this is data on DDT, chemical used to kill mosquitoes back in the 50s, 60s, and 70s, and 40s, I guess, as shown here on this birth cohort and the years at the top. Well, her important work has shown that earlier in life, these girls were exposed to DDT, the higher the odds ratio of breast cancer incidence before the age of 50 was. So you can see here the odds ratio of 5.4. Here, if they were exposed a little later in life, their odds ratio is only a 2. So there is human evidence confirming chemical exposure during development increases your risk for breast cancer later in life, and these are in premenopausal women, likely premenopausal women. But the fact that breast cancer incidence is still rising slightly in young women, especially underrepresented minority women, and the fact that breast cancer is still the #1 diagnosed cancer of women globally, or is now the #1 diagnosed cancer of women globally, is a problem. And I want to tell you about some of the efforts that we have ongoing to deal with that. So one of the things that we're doing is we're trying to add to this Hyatt model. So I and some colleagues of mine at NIEHS have undertaken have undertaken a literature review to increase the number of blue environmental factors that might be linked to breast cancer causation. And we're actually using animal data also, whereas this cancer model only uses data from women. So we're trying to increase the number of lines that are, the number of blue marks that are on this, and the number of lines that link to the blue marks. So here you can see there's not a lot of lines coming from the blue portions of this, and we want to increase that. Like, how is it linked to other things that are risk factors for breast cancer? Because I don't know if we're doing a very good job of that. You know, I think in animal models especially, we're just looking for tumors, but that's not the whole story. Like, what else about the exposure may have made those animals more susceptible? Because not all of them respond, right? Not all women respond to the same exposures. What else is making them susceptible to that response is something we need to work harder at. So perfluorinated chemicals is one example of one area that we're working in. So if you're not familiar with PFAS and living in the state of Massachusetts, I'd be surprised because Massachusetts does have its fair share of PFAS issues going on, especially due to the military bases. And so you might be familiar with PFOA and PFAS, but we are also really interested in some of the emerging replacement chemicals, such as HFPODA, or commonly called GenX. I probably will call it Gen-X. These chemicals are present in a lot of things that we have in our homes. Everyone listening to this today probably has these chemicals in their blood, and that's because you are being exposed to them from a variety of sources. Those sources include air, water, your drinking water. Hopefully in Massachusetts, maybe not your drinking water anymore, but when you participate in recreational fun things in the water, there's definitely probably exposures there. In addition to fruits and vegetables and dairy or animal products that are grown or raised off the soil or land that may be contaminated with these chemicals. These aren't very well understood exposures, especially the food products. But people like the Silent Spring like Laurel Shader and others at Silent Spring are trying to make a difference in that arena, trying to really force the hands of the FDA to regulate and measure those exposures. And but I would say, like, for the rest of what I'm going to talk about, PFAS, most of what I'm talking about is coming from industrial sites or like point source locations that are contaminating drinking water or air. In North Carolina, for example, we have very specific PFAS exposures. We have Gen-X and others that are less is even known about them than Gen-X in our water. And you can see here in the red, the proportion of Gen-X that was measured in the water, in the drinking water in Wilmington, North Carolina, when this was first discovered, compared to the legacy PFAS that are coming from a point near NIEHS and UNC Chapel Hill, where we know there are numerous landfill, wastewater treatment plant, and point source long-term contamination of the Haw River. All of this drains into the Cape Fear River, which ends up giving Wilmington kind of a mother lode of PFAS in their drinking water, which they've been dealing with for the last 5 years or so. One of the problems with PFAS is that they accumulate in the body. And one of the bigger issues with that is that they can affect pregnant women and others who drink a lot of water at a higher rate than they do the normal population. So the reason that I'm concerned about pregnant women will be clear in a minute, but pregnant women and lactating women and infants in particular drink a lot more water than do a regular population of any age. And the more water you drink, the more intake you have, and the more bioaccumulation you have of these PFAS chemicals. So, and it is known that the longer a child is breastfed, the higher their internal body burden of PFAS would be. So I'm just showing you examples from 2 studies here where by 6 months, so this is cord blood levels that were measured at birth, and after 6 months, the children have, all of the children to varying degrees had increased levels of PFOA in their blood, which leveled off after having not been breastfed any longer. And then here in this study, another study showed that at birth, here are your levels, and after lactation, and this is 11 months, they are showing significant rise in most all the women in their study for PFOA in the children. So when we study the effects of PFOA and other chemicals, we use a developing mouse model. And there's a lot of really interesting things about using the developing mouse model. There's a lot of reasons we're doing this. And that's mostly because the child and the mother have really modified ability to excrete these chemicals. But for PFOA specifically, and a couple of others, there is a big difference between the half-life, or how much is there after several days or weeks in the rat. So here, PFOA is hours and days in the rat compared to like 3 weeks or so in the mouse. And then we also don't see any sex-specific difference in the human. So although there's some variability in the mouse, the monkey, and the humans, there isn't this really dramatic, like a couple of hours in a female versus several days in a male issue. And this is because the hormones in rats regulate transporters that excrete, preferentially excrete the PFAS back out of the kidneys in the female rat, which it would be the model we'd need for pregnancy, right? So we don't use the rat for PFOA or for PFNA or PFHXA or PFHXS, some of these carboxylic acid studies, very specifically because of this excretion difference, because we want them to be as much like humans as possible. And humans don't have a male-female difference in excretion rates, and the mice don't either, for all of the chemicals tested so far, at least. So in our studies, sorry, for some reason my slides just want to move without me touching a button. Sorry about that. So in some of our first studies at high levels of PFOA, we were seeing long-term effects that resulted in, that were concerning to us. So we undertook a lot of further studies, some of which are being considered for regulatory use by the EPA right now, which is really exciting because the work by Macon et al. That we published in 2011, it was really the first to show that full exposure to low levels of PFOA caused developmental delays that were permanent. And we saw them all the way out into 18 months of age. But also very low levels caused exposure or these delayed permanent effects. And then we even went to lower exposure levels where we may have had the same exposures, but we shortened the window only around the timing of bud development for the gland, and we saw the same thing. So we've determined that the fetal bud development timing was critical for these effects of PFOA on the gland, and they were permanent. So we've recently repeated these studies multiple times now, but this is our most recent repeat, and which is not published yet, but is being submitted for publication soon, showing that here in the blue, we have a downward trend with increasing PFOA levels on mammary gland development. So normal is up here at 4, and the worst mammary development gets a lower score. Same is true with GenX, the replacement for PFOA. Here's normal, 4, up here closer to 4. And we see this decline in mammary gland development, which turns out, again, to be permanent in this study at least. But we also see it in males. So we haven't been assessing the development in males. One, because there's not a lot of mammary gland development that happens in male mice. Male rats have quite a bit more. I showed you that earlier. But the same is true. It just seems to affect the proliferation of the ducts in the males. Well, so on the right-hand side is a pubertal time point where with the control shown at the top, PFOA as kind of a control, again, a positive control, showing this delayed development. It doesn't look like the control at all. This is a lymph node here. This dark spot is a lymph node, and this is a landmark that we use for sizing and for length of the gland. It's really present in the same place in all the glands from animal to animal. So we can use that to assess how far the development of the ducts have expanded past the lymph node, allowing us to very quickly see here that not only PFOA, but a similar dose of GenX caused pretty stunted development of the mammary gland. And we took this all the way out to 18 weeks, and it's not shown here, but we do see that those developmental delays persisted not only in the whole mouth shown here, but in blinded pathology studies, we also saw these delays in development. The pathology came back with delays. This again goes back to some of the higher exposure studies that we've conducted, but I just wanted to show you that it's really the amount and the timing of the exposure that seems to matter most, because we have here controls who didn't have any exposure, and here's what the ducts and the fat cells look like at 18 months of age. But when we have only lactational exposure, which happens in women, especially in the Northeast, in the Hoosick Falls, New York area, they've reported substantial lactational transfer to children there, and intrauterine exposure in the third block. Sorry, I lost my cursor here, with the arrow showing lesion development in this structure. And then here with both in utero and lactational exposure, we see even worse development of this stromal hyperplasia. So we're seeing an increase of the amount of stroma around the epithelium, which I guess in women terms would be a more dense breast, potentially, when we have less fat and more stroma surrounding the epithelial structures, which no woman wants to have that either, as it's highly related to breast cancer risk. But we also have quite a change in the structure of the fat, as you can see here, comparing the left-hand side in the control animals and the right-hand side fat cells here in a PFOA exposed animal. And we do see these for most of the developmental window, mammary gland developmental window. We see this odd where we have more fat cells, but they're very small and unorganized. And we're pretty sure that changes the communication to these epithelial cells that are shown where the arrows are. And what's shown there is labeling of estrogen receptors. So there's definitely a decrease in the number of estrogen receptors, both at weaning and after weaning in the young adult animals, that's present just from developmental in utero PFOA exposure. But we don't see that effect on the progesterone receptor. So we don't see any changes there. It's very specifically an estrogen receptor change. How this may modify breast cancer risk isn't known. We think, so there has been a 2-year cancer study on PFOA. It did not show an increase in mammary tumors in the developmentally exposed animals. It is very possible that if those animals were exposed to a carcinogen, that may have a different outcome. We don't know, because we think these are developmental changes that might put a person at different risk for problems in the breast later in life. And that has been shown. Oh, sorry, I was going on to the next slide. I just wanted to mention here, this is a table from a recent paper by Gloria Post, where she has shown all of the endpoints that have been used for the state's risk assessments on PFOA. And Massachusetts is shown on here off to the right, because they were kind of one of the more recent additions. And so work that I was involved in from the Lau paper was used for the lowest level effects in that study. But New York has used one of our studies, this Macon study. And like I said earlier, I know it's being considered for use by the EPA also. And you know, these lead to these developmental studies, all of which are developmental studies. All have led to lower levels of regulatory or health advisory levels than the EPA currently has. And so I'm really hoping that the EPA will adopt some of these developmental endpoints that are maybe a little bit more sensitive in their ongoing risk assessment. But what I was going to say earlier was that we know now that in women, exposed to PFOA and other PFAS during their pregnancy, that the pregnant gland is also probably sensitive to the effects of this chemical, because women with the highest levels of PFOA in this study on the left, so quartile 2, 3, and 4 shown here, are all having a lower probability of nursing their child as you go out into months after birth. So there is some effect of PFAS on the duration and the amount of milk that's being produced in women with exposures. So the first quartile is shown in the blue up top. In another study from Scandinavian countries, so this one on the left is from the US, Scandinavian study on the right, showing that multiple PFAS can significantly shorten the number of months that women exclusively breastfeed, because they're not producing enough milk to sustain their child. So another set of chemicals that I want to just super quickly talk about is bisphenols. So we've taken a look at bisphenol AF and bisphenol S in comparison to BPA. They all are estrogenic and have similar, if not more estrogenic effects than BPA. And in this study in mice, using these whole mammary gland whole mounts, we were able to see lesions in the mammary gland that we were not able to see in pathology sections. So we worked with the pathologists and sectioned all of our whole mounts, and were able to confirm that quite a few lesions were missed in the pathology single section screening of the glands from this study. And there was a significant increase in the number of problems demonstrated and diagnosed in this study using sectioning of the mammary gland whole mounts. So one, there's probably a difference between the right and left-hand side. You know, it's the same in women. You don't form a tumor in both left and right at the same time necessarily, maybe one or the other. But there's a lot of tissue there, and if you're not looking at all of it, you may be missing something. So using this whole mount approach, we were able to look at much more percentage of the breast or the mammary tissue than we would be in a single section pathology. And we were actually able to diagnose adenocarcinomas within the glands, shown here on the bottom. So one of the things that, in closing, I want to just bring up some discussion points. One of the things we need to do is address gaps in knowledge in a more thorough way. So changes in breast developmental timing need to be addressed better for environmental effects. This issue of shortened duration of lactation, shown here on the left, is really an issue that has gotten very little attention. There's really 2 studies in the last 20 years that I know of that have addressed this issue. Both of them happen to be on PFAS. There are studies from longer ago than that have touched on this, but these 2 studies are unique in that they've incorporated women who didn't know they had the exposures, so they could not have changed their mindset on wanting to breastfeed. These women were determined to breastfeed. So this was really set up like I'm going to breastfeed my child and there's no doubts about it. But then the investigators measured the PFAS and later were able to determine that it affected the ability of those women to breastfeed without their knowledge. So there's no bias here in terms of the environment. Again, with the breast developmental timing, the same could be said there. You can't change that biology. You don't know that it's happening, so you likely would not be able to change your exposures because you don't know it's happening. So there are other breast endpoints that I think we should be looking at to gain knowledge about the environmental effects. And there's this other issue that's recently been brought up by the Cone group, again, Mary Beth Terry and others, showing that we actually might be thinking about environmental causation in the wrong generation. So in their study that was recently published, and that it's shown here as F2 study, they looked at DDT levels in the pregnant mom and looked at multiple endpoints over time. So I've already talked about breast cancer in women under the age of 50. I didn't talk about this, but they've also shown that there's a difference in breast density and breast cancer here. Oh, sorry. I didn't talk about the dark blue one. I did talk about the gray study where there's a change in their offspring's breast cancer risk under the age of 50. But now they also have shown that the granddaughters have earlier puberty, they have earlier menarche, and they have a change in their BMI that's related to their grandmother's BMI level. So the grandmas are the ones that had the exposure. And so both menarche and obesity are risk factors for breast cancer. Whether or not these granddaughters go on, these are daughters, yes, granddaughters. Whether these granddaughters go on to also form higher breast cancer risk, or higher breast cancer risk is not known yet, won't be known for 2 more decades, really. But they're able to attribute these breast cancer risk factors to grandma's exposure to DDT. Now, whether or not we've been thinking about this wrong all along, we used to look for the key under the lamppost back in the 80s or maybe early 90s, and now we know to look in utero exposure. Well, maybe we need to look further back than that. I think there's some real thought that should be given to this, and this is something that's been brought up in the NCI, NIH discussions that are happening, so that we can really start to think about this in terms of study design. So animal studies would be a great place to really think about testing this hypothesis. And then, in this situation, for obesity and early menarche, we definitely might be looking for causation in the wrong generation. One last note. You may notice a couple familiar faces on this page, but Anna Soto from Tufts, Sophie Christensen, Laura Vandenberg from UMass Amherst, Julie Boberg from Danish Technical University, Ruth Ann Riddell from Silent Spring Institute, and her trainees that are working with her, Jenny Kay and Besaida Cardona, and Linda Birnbaum and I are all working together to try to add mammary gland evaluation to some of the OECD testing, so that one, we can really provide some mammary evaluation methods that everyone can use. And we also want to increase the amount of knowledge that we get back from every test guideline that's run on anything that might be an endocrine disruptor. So we're trying to get like the US and Europe to change the way they collect and evaluate the mammary gland. Right now, they really don't. So it's important to do this because of the health effects that I've talked to you about, the public health urgency. But really, we're not doing a good job. I say we. I might be doing a good job, but in general, as a testing universe, we are not doing a good job of mammary gland developmental screening for effects, whether it be puberty, lactation, tumor formation, inflammation, whatever it may be, permanent developmental effects. We aren't doing a good job, and these are some things that we're working on, and this is probably going to take us a couple years to finish and to really push this ahead. But these are some of the things we're doing to really shift gears and increase the momentum in this area. And with that, I'll take questions, but I just wanted to also thank the people that I've collaborated with on a lot of these projects over the years, and the people who've worked on the projects are noted at the bottom. Thank you.
Thank you, Dr. Fenton. So if any of the audience have questions, please submit them through the app. We did have one question just for clarification on what Gen X means.
Gen X is the nickname for HFPODA, or it's the replacement chemical for PFOA. So they've taken, PFOA has kind of come off the market in the US and in Europe, and Gen X has replaced it.
All right. So we do have another question. How do you decide which chemicals to study? Do you base it on questions from concerned communities or other factors?
Right, really good question. So we are, in my lab, we try to focus on ones that are either going to be undergoing review for risk assessment or maybe high use chemicals so they're either a lot of people are being exposed to them, or potentially they're replacing chemicals where a lot of people were exposed to them, like Gen X, for instance, or atrazine, for example, where there's already some regulatory, it's already regulated. And so we were hoping to decrease the regulatory level for that chemical. So it varies, but usually it's because there's either something coming up on the horizon that we want to affect, or we know there's a lot of people exposed to it. So for example, we would really like to study the other chemicals that have no health effects data that are in the water in the Wilmington area, but we actually can't get our hands on those chemicals yet. There's a moratorium on production of those chemicals by some of the industry folks that make chemicals for sale so that we can use them in testing. And there's been a lawsuit that stopped them from producing them, so we can't buy them right now. So as soon as we can get our hands on them, we'll test more of those too, because we know there's a lot of people exposed to them. We just don't know anything about them.
So another question we have is, do you know what causes some chemicals to cause delayed mammary gland development versus precocious mammary gland development?
Right. We don't know exactly, but we have studies ongoing right now using transcriptomics that will help us get to the answer for that. So we have, we've compared the gene pathways that are being altered, and what we know in the developmental delays, we think that we're actually affecting the stromal epithelial communication. So there's more stromal, it's promoting the stromal tissue to stay there more than the epithelial growth. So it's very complicated. We see these epithelial mesenchymal transition pathways are being regulated. And so we know that it's the communication between the epithelium and mesenchyme, or those stromal cells, that are really being altered, in addition to growth factors that are needed for the outgrowth. In the precocious development, we don't see the effect on the mesenchyme like we do in the delayed development. So we think it's completely different pathways that are being triggered. And some of the pathways being triggered for precocious development are those that are estrogen-related receptors, ERR alpha, ERR gamma, and they're kind of they can promote estrogenic signaling, but they're not estrogen receptors. So I think there's some rethinking that may have to happen in terms of our thoughts on estrogen or estrogenicity. I don't think it's just through the ER. I think there are also effects through estrogen-related receptors that need to be considered.
So you had mentioned that the BPAs were estrogenic, but a lot of the other chemicals are not.
Right, right. And honestly, Other labs have tested chemical, like we've tested nonylphenol, and there haven't been a lot of other estrogenic chemicals that have been tested. And I think one of the bigger issues is the progesterone related chemicals. I know there are chemicals that act as progesterones because we've seen that in like some of Susan Engel's work on fracking material. So we know there are chemicals in the environment that people are exposed to that act like progesterones. And I feel like those are also very important and have not been had enough attention paid to them, because like for hormone replacement therapy, we know that the HRTs that were both estrogen and progesterone led to the worst outcomes in women. And once people stopped recommending them for women, there was this decrease in breast cancer incidence. So I feel like there are still some really unanswered questions there. And honestly, we've just kind of touched on the tip of the iceberg, I think, really, as far as estrogenic chemicals.
So kind of as a follow-up to what you said a minute ago, so almost all breast cancer is epithelial. And a lot of the time we look at the development of a breast cancer as going from epithelial hyperplasia to atypical hyperplasia to in situ cancer.
Right.
So how does that process fit in with the hyperplasia not necessarily being related to the carcinogenic effects?
Right. Well, there is some evidence, both in some studies of mine and in studies Anna Soto has conducted, of these intermediate kind of pathological lesions. So what might be called atypia in women, and some ductal carcinoma in situ in women. We do see some of that in the rodent mammary glands. And so I think there are chemicals that can act as a carcinogen. So they can initiate the problem and they can promote the problem, leading to a tumor. BPA is one of those. But then there are probably others that have different modes of action, whereas they may change the stromal composition, they change the microenvironment. Those chemicals may need a second hit or a carcinogen to actually see the potential for them to cause cancer. Like what would happen in a woman? We're exposed to a variety of things throughout our whole life, right? Animals aren't. So if we don't expose them to a second hit, we don't see that. So there are some testing issues where we could do a better job. And I think that goes along the lines of some of the things that myself and my cohort of colleagues are trying to push this forward. Like we need to think about this differently. We need to ask those questions that you just asked me. Like, why isn't there, the reason this isn't translating is because the study wasn't done to be able to translate it. In some cases, we can translate it, like for bisphenol A. We see some of those intermediate, and we see adenocarcinomas form, both in GLP-like studies, like the one that was the Clarity study that was conducted. We saw tumors in the lowest dose group there, in the low dose BPA, which is where the academic studies have shown effects of BPA. And in smaller studies like my own, where we don't run it under GLP and we are doing this in mice, not rats, we are definitely seeing those intermediate lesions.
Right. So I have another question, but again, I'll just remind the audience, if you have questions, please submit them through the app feature. So the whole breastfeeding lactational part seems a little bit counterintuitive to me. So if I understand correctly, you're saying that a lot of women who've had PFAS exposures are producing less breast milk, but yet the highest risks come from the longer duration of breastfeeding.
Well, yeah. So unfortunately, breast milk is a really great way for women to get rid of their body burden of PFAS chemicals. And that's true for others too, like brominated flame retardants and some other lipophilic chemicals. But so quite a bit of what the mom has in her will transfer to the baby through the milk. So the baby's body burdens go up, but mom's burdens go down. But what has happened for this breastfeeding duration has happened during pregnancy, not during lactation. So during pregnancy, the breast has to go from this kind of normal wispy ducts, like I showed you in a lot of the pictures, to a very full gland full of milk and full of epithelium, where you really can't see the stromal or the fat cells anymore in a pregnant lactating gland. So it's full of epithelium. So we have shown in animals, and I didn't show you this data, but we've shown in animals that PFOA actually prevents the filling of the gland with the epithelium that makes the milk. So in the women who had shorter breastfeeding duration, I'm hypothesizing that they probably have less epithelium producing less milk in their breast. Whereas a woman who didn't have as high of an exposure likely has more epithelium and therefore can make more milk. So that's really where this is coming from. The effect of PFAS, PFOA in this case, happened during pregnancy and not during lactation. But you see the repercussions of it during lactation.
All right. Are there any other questions from the audience? All right. It's just about 1:30, so I guess we'll wrap up. On behalf of Cheryl Osimo, MBCC's Executive Director, and the rest of our Board of Directors, I want to thank Dr. Fenton for this very informative discussion today. I want to thank our listeners for joining us. For those interested, the recording of this webinar will be made available later today on the MBCC website at www.mbcc.org. So thank you, everyone, and have a good afternoon.
Yeah, thank you.
Thank you, Dr. Fenton.